Like a post-star equities.
Well, I mean,
so, thank you everyone for being here this afternoon. It's a beautiful day out there. Those who haven't been there, we still can be. But I'm really thrilled to have a friend for many years now, Professor Jalife, who is now as the distinguished M. Catecholaminergic at the University of Michigan, and he heads the Center for Arrhythmia Research at the University of Michigan. It's a recent move from the University of SUNY Syracuse. Dr. Jalife really doesn't need an introduction. I mean, everybody here knows his papers, his work, his books, his contributions to the field are enormous. And he still continues now in this new place to chart new directions and territories. So he got his MD degree in Mexico City, in Mexico, and then I think had some clinical training in Madrid. And after that went to SUNY Syracuse, where he did a postdoctoral fellowship, and at the same time worked in Gordon Moe's laboratory at the Masonic Institute, which was one of the main centers for cardiac arrhythmia research. And he then stayed in Syracuse as the chair of the Department of Pharmacology. Since then, the pharmacology department there had some other aspects, but its contributions to arrhythmia studies from Dr. Jalife and many of the researchers who work with him are really a basis for many, many of the things that I've done everywhere, including here. So, thank you for coming, and it's all yours. You'll tell us about CPVT. You'll hear what CPVT is in a minute.
well, Yoram, thank you very much. It's always great to see you, and I'm always honored to be introduced by you, and it's great to come here, which is a mecca for electrophysiology, cell biology, ion channels. I really am delighted to be here. And what I would like to tell you about is work that we did a couple of years ago, and then some new work that is cooking right now in the laboratory, and that I hope that you will find interesting. Catecholaminergic polymorphic ventricular tachycardia is a very rare disease, but I do think that it can tell us a lot of things about other types of disease, particularly heart failure, in terms of the mechanisms of sudden cardiac death. And I hope that what I will show to you today will have you agree with me that this is could be of interest, not only to those who are interested in inherited arrhythmias, but those who are interested in cardiac electrophysiology, ion channels, and also sudden cardiac death. So all of you know that heart failure affects many people in the United States, and it's associated with high mortality primarily because of the contractile dysfunction associated with pump failure or with sudden cardiac death caused by ventricular arrhythmias. And these ventricular arrhythmias are really of unknown origin. What initiates these arrhythmias has been studied by a number of investigators, but I will show you that, to my knowledge, the origin of these arrhythmias has really not been established. Now, the fact that patients develop contractile dysfunction or sudden cardiac death are not mutually exclusive. In fact, those who develop contractile dysfunction many times die suddenly as a result of arrhythmias. And the genesis of heart failure syndromes is very complex. You all know that is multifactorial, but cellular calcium alterations and its regulation may be the final common pathway in both the pump failure and the arrhythmogenesis. And I've just listed a few of the prominent papers that have been published in relation to this problem. And I want to bring your attention to the studies of Steve Polwitz, who really has done, yes?
No, he used to be here.
Oh, okay. So you all know that Steve really has been one of the major contributors to the field. Okay. So just a reminder of excitation-contraction coupling. It took me a long time to make this slide, so I hope it works. Okay. So excitation-contraction coupling involves a bunch of proteins that include, obviously, the sodium-calcium exchanger, the calcium channel, the calcium ATPase, the ryanodine receptor, the circa-2 pump, phospholamban, and the calsequestrin, as well as the contractile proteins. So when an action potential comes and activates the cell, calcium channels enter through the L-type calcium, enters through the L-type calcium channels, and that activates the release of calcium from the SR, and that calcium moves to where the, not bad, huh? I'm really proud of this. Moves to where the contractile proteins, where it combines with troponin, which normally exerts an inhibitory action to myosin acting interactions. Then the myosin heads go and combine with the actin, and that initiates the contraction. Relaxation occurs because calcium is pumped back into the SR, or it moves outward through the various pumps and exchangers, and that makes the contractile proteins move down and relaxation occurs. So normal excitation contraction coupling is a result of calcium signaling. And calcium signaling is a major factor in determining the contraction. And so, in fact, in normal conditions, in an electrically paced myocyte, in the presence of a calcium dye, you will see that there's uniform calcium elevation, and there will be a one-to-one relationship between the calcium release and the contraction. And this can be demonstrated using laser scanning confocal microscopy, as shown by this slide and by these movies. So every time there's calcium release, and that calcium release occurs more or less homogeneously throughout the cell, there will be a contraction. And the relationship between the calcium transient and the force is always one-to-one under normal conditions. Do you move? Okay. So what happens in heart failure? Heart failure is associated with remodeling. And there are several things that happen. One of them is the circa-2 pump gets downregulated. The sodium-calcium exchanger paradoxically gets upregulated. The ryanodine receptor becomes leaky, and this is the subject of today's presentation. And the contraction of the heart goes down. And this is going to be the focus of our presentation, primarily because during heart failure, there is a problem with excitation-contraction coupling. The release of calcium is no longer synchronous throughout the cell, but it becomes very asynchronous as a result of the presence of non-uniform calcium transients, and calcium can be elevated when it should be at rest. So in this movie, you will see that indeed in the failing myocytes, the calcium release can occur at different phases of excitation and the diastolic interval, and this can occur not homogeneously throughout the cell, but there can be calcium release at times that are out of sync with the normal excitation-contraction coupling process. And this calcium release can produce delayed after depolarizations. Particularly, you can induce these by repetitive stimulation, the state action potentials, and then after a period of rest, you will see an oscillation of the membrane, and this oscillation of the membrane is produced by the release of calcium. This is in the presence of caffeine that will eventually, at large concentrations, will induce delayed after depolarizations. And as demonstrated by Steve Powitz and his group, when he was collaborating with the Emory group, they demonstrated that indeed the threshold for the DAD formation, it occurs at a much lower calcium concentration in the failing heart than in the normal heart. So, ventricular tachycardia in non-ischemic heart failure it's initiated by a non-reentrant focal mechanism that appears to be due to triggered activity, primarily from delayed after defluorizations. And this is the main message that I want you to remember for the rest of the talk. The experiments of Steve demonstrated that in a rabbit heart failure model produced by aortic insufficiency and constriction, he was able to demonstrate that the arrhythmogenesis in this heart failure model is primarily focal. You can get focal ventricular tachycardias, and these focal ventricular tachycardias are thought to be associated with delayed after depolarizations. And there is no question that these delayed after depolarizations and these foci were present. The problem is that we really do not know where those foci start. And that's the major message that I wanted you to get. So, focal arrhythmia seem to be true for 100% of the ventricular tachycardias in human non-ischemic cardiomyopathies, and in 50% of those who suffer from ischemic cardiomyopathy. However, while the ectopic activity that initiates these arrhythmias in heart failure is likely to be focal, its precise origin has not been elucidated. This is when CPVT comes to the picture. CPVT is a rare inherited arrhythmogenic disease that is characterized by adrenergically mediated, that is, exercise-induced rounds of bidirectional tachycardia, polymorphic ventricular tachycardia, and this leads to syncope and or sudden cardiac death in the absence of structural heart disease. So, a rare arrhythmia induced by exercise that occurs in young people, that is inherited, and that is present in the absence of heart disease. And it's accompanied by very peculiar arrhythmias, bidirectional tachycardia, and polymorphic ventricular tachycardia. These are the clinical characteristics of those patients. It is a very lethal disease. It's about 30% by age 40. The heart is structurally intact. There is no electrocardiographic abnormalities at rest, and the tachycardia can be induced during exercise, and it's what establishes the diagnosis. The types of arrhythmias, again, are bidirectional ventricular tachycardia, polymorphic ventricular tachycardia, and VF, and sudden cardiac death is sometimes the first manifestation of the disease. Beta blockers are preferred in these patients, but unfortunately, they do not seem to prevent the sudden cardiac death. And finally, ICD implantation is of choice of these patients who are not adequately protected. So you can imagine that you have to put an ICD in a little young guy of 4 to 5 years of age. That's a difficult decision for the cardiologist, and it's a very difficult problem. This is just a Kaplan-Meier curve showing that about 30% of CPVT patients died before reaching age 40 years of age. And for some reason, which I don't know what the mechanism is, males seem to be more effective than females. They have to do with hormonal basis. You can propose any hypothesis and it'd be reasonable to test. And this is the characteristic arrhythmia that you see in patients with CPVT. These are 5 ECGs from 5 patients that Silvia Priori, who was the scientist who described this arrhythmia, the genetic basis of this arrhythmia obtained. The most interesting aspect of this is that if you look at the QRS alternates on a beat-to-beat basis 180 degrees. Not only that, but the arrhythmia is associated with a narrow QRS. And so those are the two major characteristics. The tachycardia is not a very fast tachycardia, and these patients, while they have the tachycardia, can still function. But the problem is that it deteriorates into polymorphic ventricular tachycardia and ventricular fibrillation. And this can be induced by an exercise test. You put the patient on the treadmill and they start exercising, they develop their bidirectional tachycardia, polymorphic tachycardia fibrillation. Many times they go directly from normal sinus rhythm to the ventricular fibrillation.
Okay. Would they have activation of one half of the ventricle and blocking the other half, and then activation of the second half of the ventricle, the next complex?
You want to preempt my...
I'm sorry.
Yes, that's what it is. That's what we think it is, but that we don't know. We didn't know what the mechanism was until we started studying this arrhythmia. I will tell you exactly what we think is the mechanism of the arrhythmia itself. And that was the subject of our work. So, that's what we wanted to do, unravel the mechanism of the bidirectional tachycardia. And we thought that this may help understand mechanisms of arrhythmias in heart failure. Patients with heart failure may develop bidirectional tachycardia, by the way, but they develop them primarily when they are given digitalis. When you give a patient with heart failure digitalis, bidirectional tachycardia appears. So it seems to be related to the calcium overload mechanism.
You give people with heart failure adrenaline, which you wouldn't do.
Right.
Will that induce it in other heart failure patients? Just like
In exercise in heart failure,
give them catecholamines.
I wouldn't give catecholamines.
No, you wouldn't. But are you sure it's not the same thing going on?
Well, we don't know. That's but I want to know. I don't know what the focal arrhythmias in heart failure are, so I'm using this as a model to test for a mechanism. Now, CPVT is caused by mutations of genes encoding for two major proteins responsible for the control of intracellular calcium: the ryanodine receptor type 2 and the calciquestrate 2 protein. We focused our attention on the ryanodine receptor, but any of these two can result in the phenotype of catecholaminergic polymorphic ventricular tachycardia. Okay. Any of those, which takes the culprit back to the release of calcium from the SR. That seems to be the final common pathway of the origin of the disease, which is similar to what happens in heart failure. So the genetic basis of the CPVT and the RYR2, there are approximately, so far, 70 point mutations have been described in the ryanodine receptor type 2 of the human heart that are associated with this disease. And these mutations result in functional defects in the calcium channel of the sarcoplasmic reticulum, and this leads to intracellular calcium overload. And similar to heart failure, the mutations lead to calcium leakage from the SR, and this results in membrane potential oscillations, and therefore, the CPVT patient has arrhythmias. Again, in the patient with CPVT, DAD seem to be the major culprit of the arrhythmias that lead to triggered discharges, which eventually result in the bidirectional tachycardia and the monomorphic ventricular tachycardia. But here we're just dealing with generalities, as you very nicely pointed out before. So this mutation was the first mutation that Silvia Priori described in 2001. And this mutation was discovered in an Italian family which demonstrated a highly malignant phenotype. This is the pedigree, this is the family. You can see that the penetrance in this family was really very important. And this is the proven where the mutation was discovered, an arginine to cysteine mutation. And this was sufficient to generate the phenotype of the CPVT. This particular mutation is highly life-threatening. It occurs in about 50% of patients. The arrhythmias occur in about 50% of patients despite the beta blockers. And in vitro models using expression systems initially demonstrated that the mutation results in increased release from the SR, which led to the speculation that the arrhythmias were associated with abnormal calcium release. But how do you link the mutation to the bidirectional tachycardia? That is the question that we wanted to address. So the objective was to determine the electrophysiological mechanisms of arrhythmias. And we took advantage of a unique knock-in model, a mouse model that CBAC made. And this had the mutation, the murine equivalent of the human missense mutation. This is the R4496C in the mouse, which is equivalent to the R4497C in the human. And this is a remarkable similarity between the arrhythmias in the patient and the arrhythmias in the mouse. I showed you this slide before. This is a patient with CPVT that underwent exercise and developed the bidirectional tachycardia, which eventually deteriorated into polymorphic tachycardia and sudden death.
You know, Pepe, this is like one of those mysteries on television where you're going into, I think I understand, I think I solved the problem.
Okay.
It's not clear, but I think I understand.
You solved the problem. I knew that you would.
I know you.
So anyway, in the mouse, you can exercise the mouse and try, but it's much more difficult to exercise the mouse than give it caffeine and epinephrine to induce the arrhythmias. And this is the bidirectional tachycardia. And again, you have 180-degree changes in the QRS, polymorphic tachycardia, and eventually ventricular fibrillation with a bolus of caffeine and epinephrine. So all the evidence supports the idea that these arrhythmias in these mice are caused by enhanced calcium release through the RYR2 channel. And indeed, if you take a ventricular myocyte from this mouse, well, one of these mice, what you will see is that under resting conditions in a wild type, if you apply a train of repetitive stimuli and then let the diastolic interval persist, you will see no oscillations in the membrane, even if you give it isoproterenol at 30 nanomoles. But in the RYR2 mouse, even in the resting condition, you start seeing delayed after depolarization, and when you give them isoproterenol, you will get even a tachycardia development in the ventricular myocyte. So, yes, the electrophysiological phenotype that you see is associated with delayed after depolarizations and triggered activity. But what is the mechanism of the bidirectional tachycardia, and how does this bidirectional tachycardia eventually lead to sudden cardiac death? Now, I want you to pay attention to one fact. First of all, the QRS changes 180 degrees. And the best way of looking at this change in the QRS would be from the right proportion leads, E2, E3. And you will see that indeed the QRS changes. The second, the QRS complexes are relatively narrow. And this is just a cartoon of a QRS and shows that indeed the QRS is not very wide compared to the normal. You know that the normal sequence of activation starts in the sinus node, spreads through the atria, goes to the AV node, and then goes through the Purkinjes, and then to the muscle. So this normal sequence of activation is what brings this narrow QRS. So it seems like the specialized conducting system might be involved in the mechanism of this arrhythmia. And it really has been a forgotten link in many of the arrhythmogenic diseases that people have looked at, including long QT syndrome, heart failure, etc., etc. Was this a mystery that you wanted to solve? So, I remind you, normal sequence of activation, sinus node, AV node, his bundle, left bundle branch, right bundle branch. Notice that the first activation of the ventricles occurs from endocardium to epicardium. And the first activation actually occurs in the free walls of the right ventricle, the free wall of the right ventricle, and the free wall of the left ventricle. So, if you want to look at the outside of the heart, what you will see is breakthroughs that appear right on the epicardial surface, on the lateral surface of both ventricles. And this was very nicely demonstrated many, many years ago by Dirk Durer and his group, Giel Janse and all of those guys in the Netherlands, who took human hearts and isolated them, did Langendorf perfusion, and did multiple electrode mapping of the epicardial surface to show that indeed you have a breakthrough in the right ventricle that is followed very rapidly by a breakthrough in the left ventricle, and then the activation moves toward the base of the heart. And these are just two different hearts showing that the sequence of activation is almost identical in all of the human hearts. Now, let's see how this could relate to bidirectional tachycardia. So we use a very simple three-dimensional mathematical model of the mouse ventricles. Actually, this was a dog heart that was just scaled down to a mouse size. That included the His-Purkinje network. And it was a very primitive His-Purkinje network, but nevertheless, it helped us think this model. So the normal sequence of activation of the ventricles, this would be the right ventricle, this would be the left ventricle, and you are making a section of a longitudinal section of the heart. And we don't have atrium, we don't have AV node, but we have the His bundle, and we're going to pace the His bundle, and then look at the normal sequence of activation that produces these repetitive narrow QRS, looking at it from the endocardium. And what you will see is a very nice rapid activation of both ventricles with excitation of the both branches of the His-Purkinje system with excitation of the both ventricles. And when then you look at it from the epicardium, what you will see is the breakthroughs. And these breakthroughs, which will appear in a second, they, are almost simultaneously appearing on the right and the left ventricle. And this is what you will see, similar to what Dirk Durer saw in the human heart, and you will have a narrow QRS. So what happens if you now have two foci in the His-Purkinje system, one in the right and one in the left of His-Purkinje system? And these foci are in the ryanodine receptor mouse. It's a prediction, which has been mutated, and these foci actually are capable of generating delayed after depolarization. Well, if you now get the excitation on the right, on the left, you will see activation from left to right. If the foci now of the left, of the right fires, you will see activation from right to left. And this would give you the 180-degree change in the QRS with bidirectional tachycardia. Yes, sir?
But the QRS would have to be wide. You've got a mouse model that says we could be narrow anyway. But
yeah,
if it propagates, in that direction, like bundle branch block, then it's got to be a wider QRS than normal. So there's something else has to be going on for the QRS to be narrow.
Right, but the QRS is not absolutely the narrowest QRS, neither in the mouse nor in the actually there is a bundle branch growth configuration in both. but it is not the white QRS that you see when you have a ventricular, distal ventricular activation. So what we are proposing is that perhaps the bidirectional tachycardia has to do with focal discharges in the His-Purkinje system that are alternating on a beat-to-beat basis. That is the hypothesis that we wanted to test. Okay, and that will give you the configuration of the alternating QRS. So where is the origin of the bidirectional tachycardia? Is it in the Purkinje or is it in the muscle? So can we do experiments to determine that question? Well, first of all, we want to know that mouse does have Purkinje fibers. And until recently, nobody had described the His-Purkinje system in the mouse. So we went on and did some histological sectioning and made a collage of the endocardium of the right ventricle to show that indeed the His-Purkinje system is almost identical to that His-Purkinje system of any other mammalian heart. And it has a His bundle, it has the bundle branches, and it has all of the branching network that characterizes the Purkinje. Not only that, but at certain points, it goes and the Purkinjes attach to the muscle at the Purkinje muscle junction. This is a Laxi stain that stains the Purkinjes. Okay. Now, we know that there are Purkinjes, so what we did is do optical mapping experiments in normal and mutant mouse hearts. We perfused it in a laminar perfused system, and we used a CCD video camera. We usually use the side measure camera in our laboratory. We use a potentiometric dye 4NFs that enable, I don't have to explain that technology here. You all know what it means. And some animals receive, some hearts receive epinephrine, and 7 out of 10 develop arrhythmias. And in others, we gave calcium and isoproterenol, elevated calcium and isoproterenol, and 6 of those 9 developed the arrhythmias. And this is the system. We use a dual video camera system that enable us to detect the fluorescence in the red spectrum, and this we use somehow to eliminate the motion artifact. We didn't use the blebistatin or any of the other electromechanical uncouplers. So the first thing that we wanted to know is does the mouse have the same sequence of activation as the human heart when you look at it from the epicardial surface. So the idea was to just put the camera in front of the anterior wall of the ventricles. This would be the left ventricle, the right ventricle, the septum would be right here, apex and base. And we just wanted to look at normal sequence of activation during sinus rhythm. And what we saw was indeed the presence of 2 breakthroughs, one on the right ventricle, one on the left ventricle that activated the entire heart. So we could be sure that here we were looking at the His-Purkinje system giving rise to the normal sequence of activation. And this is just the still images showing the isochronal maps showing the right activation, left activation, and then propagation toward the surface. And we were able to show this to happen during sinus rhythm and also during atrial pacing. So everything seems to be working fine now. Now, what happens during bidirectional tachycardia? This is in Spanish. This is a mutant raton mutante. This is a mutant mouse. This is the heart, left anterior descending artery, left atrium, left ventricle, right ventricle. During normal sequence of activation at rest, no catecholamines, no caffeine, we get 1 breakthrough on the right, 1 breakthrough on the left simultaneously, and that leads to the normal excitation of the ventricles with normal sinus rhythm. We give 2.7 millimolar calcium and 100 nanomolar isoproterenol, and lo and behold, we get a bidirectional tachycardia that is indistinguishable from that of the model or that of the mouse in vivo, and you get left activation, right activation, left, right, left, right, left, right, left. And you can see that the epicardial activation is occurring very, very low, but the fact is that that activation is coming from the system, we think. So, bidirectional tachycardia seems to be a bifocal phenomenon in which two highly localized ectopic foci, one in the right ventricle and the other one in the left ventricle, mutually synchronize. They mutually synchronize somehow. We still do not know how, but they do synchronize and they discharge in an alternating fashion. So the question that we are asking, are the two foci in the working ventricular muscle or in the Purkinje system? The possibility exists that they are both on the epicardium, and they are alternating somehow, mutually entraining each other. Now, before we go and test out, does the sarcoplasmic reticulum of the Purkinje cell in the mutant mouse leak calcium spontaneously? We do that. The myocyte does leak, but does the Purkinje leak? And so this was the difficult part of the experiment. What we needed to do is isolate the Purkinje cell from the mouse. And I can assure you that's not an easy thing to do. So what we did, we made some comparisons in single isolated cells, wild type and mutant mouse. We used two models. And initially I want to show you a model that really facilitated, made our lives so easily that we were able to isolate the Purkinjes. And we just looked at differences in intracellular calcium concentration and also electrically induced spontaneous calcium release activity. Okay? What we used was this model. So this is a GFP mouse that is driven by the connexin 40 promoter. And that enables you to visualize the Purkinjes with fluorescence. And so this is a real image under the fluorescent microscope of these Purkinjes. And this model was made by Micro et al. In France. And she was kind enough to let us use the mouse. So we crossbred with the mutant mouse, and we're able to look at the Purkinjes, isolate Purkinje fibers, dissociate the Purkinje cells, and study their calcium properties. So because they were fluorescent, the Purkinjes were able to differentiate. This is a bright field image. We were able to differentiate muscle cells from Purkinje cells. And Purkinje cells have a very specific configuration. First of all, they don't have teeth to use. Second, they are thinner than the muscle, than the ventricular myocytes, and they're elongated. They're spindle-shaped in the mouse. And you have here one Purkinje. This correlates here. You have another Purkinje, and you find one right there. So wherever you have fluorescence, you will have a Purkinje cell. And you can pick up that Purkinje, put a patch pipette, and look at the ADs, or you can place it under the microscope and look at, load it with a calcium dye that does not interfere with the fluorescence of the GFP, and put it under these conditions and look at contractility and calcium measurements. And this is using this ion optics technology. And this software and everything was developed by Joe Metzger laboratory. Todd Heron was responsible for this technology. It really works beautifully with images. So this is the protocol. Use epifluorescence for calcium imaging. Pace the cells, yet whether a Purkinje or a myocyte. These are field stimulation at 5 to 6 hertz. Then pacing stop and calcium concentration or contraction is continuously reported. And then analyze parameters of spontaneous calcium release, and these included the number of and the event frequency, the spontaneous calcium release amplitude, and the latency of the first event post-stimulation. So here are examples of wild-type parts. This is during rapid electrical pacing. Post-stimulation, look at the calcium and there is no oscillatory changes. Nothing else happened. These are calcium transients that occur during the stimulation. The events in myocytes and Purkinje cells in this mouse demonstrated that indeed after stimulation, we stop the stimulation right here, you do get an oscillation in the ventricular myocyte, but nothing else happens. You do exactly the same thing in the Purkinje, and this is what happens. You see oscillation, you see all sorts of releases, and it reminds me of some of the nerve oscillations that are seen in bursting cells. Very interesting. But these are Purkinje cells of the heart. So this is just a quantification of the number of events, the frequency of events, significantly different in the Purkinje and the muscle. This is the quantification of the amplitude of the fluorescence and the duration of the events, significantly larger than the Purkinje. But something, and this is the spontaneous contractions in the ventricular myocytes using just looking at the sarcomere length, and what you find is that in the ventricular myocyte you do see spontaneous contractions after the termination of the stimulus. In the case of the Purkinje, you see many spontaneous contractions, and these very interesting oscillatory activity in crescendo that occurs many, many times. So, but something may be happening as a result of the GFP. I mean, what if the GFP is altering things? So we decided to test the same in the absence of GFP by just looking at the morphology. Is the presence of the GFP affecting the results? I mean, this was for the reviewers.
We're not here.
Okay. So can I skip this?
Probably.
So can we differentiate between isolated Purkinje cells and ventricular myocytes in the absence of GFP? We can. We do know we did some electron microscopy, and clearly, while there are organized myofibrils in the Purkinjes, they are not as well organized as those in the muscle. So if you do histology in these Purkinjes, they're totally different from the ventricular myocytes, so they don't have the D2.
What's the rest of that? Mitochondria?
What? These?
Yeah.
No, these are
No, no.
myofibrils.
No,
Oh,
the
these
rest
are
of it.
mitochondria. Oh, yeah. Maybe some SR somewhere. One very interesting aspect, if you load them with a calcium indicator, in this case fluid 2, there's no GFP here. This is fluid 2. The Purkinjes are glowing. Even the resting Purkinjes are glowing compared to the ventricular myocytes. Just amazing how different the amount of free calcium in these cells. And in the mutant, not only they're glowing, but they're sparking. It's just unbelievable the amount of calcium that they have free in the cytoplasm. And indeed, we just quantified the cells, and the cells are wider, the ventricular myocytes are wider, Purkinjes are thinner, and now we are quantifying the length of the cells also to make the story complete. So we can differentiate, even without the fluorescence. We can open the ventricles, isolate Purkinje cells, look at them under the microscope, and identify the Purkinjes compared to the muscle cells with relative ease. So now, we do the same experiment, and again, repetitive stimulation, stop the stimulation, and this is the spontaneous calcium release in the Purkinjes, spontaneous calcium release in the ventricle, but this is just an enlargement of those events, and indeed, there's significantly larger in amplitude, and their frequency is faster, and again, we quantified this, and there was no difference with the GFP mouse. So we can be absolutely sure that there's a significant difference in the properties of the Purkinjes between with those of the muscle. And also the latency for the events. This is in the ventricular myocyte. The latency for the event is significantly lower than the latency for the first event for the Purkinje cells. And I'm sorry, I said it exactly the opposite. Purkinje is lower than the muscle. This is muscle, this is Purkinje. So the latency is shorter for the Purkinje.
Excuse me, Dr. On the previous slide, was there a difference in the scale in the mutant and the wild type?
Difference in the scale. You mean?
The number of events. So the wild type is on the left and the mutant is on the right?
Yeah, no, I think it's the same scale. This is just,
Purkinje versus, okay.
yeah. This is Purkinje. This is event frequency, and this is the number of events.
Okay.
Right? And this is Purkinje, and this is muscle. Okay. So we know that calcium release in the Purkinjes is larger, occurs more frequently, the latency is shorter, and so do the Purkinjes develop DADs? Well, of course they do. And this is a normal Purkinje, wild type, in which we have given caffeine plus adrenaline, stimulated at low frequencies, 1 hertz, 5 hertz, 10 hertz, you get one small DAD. But if you give the caffeine plus adrenaline, you will get larger amounts of DADs, as you would expect from Purkinje cells. But in the mutant, all hell breaks loose. I mean, you just don't have to give it the adrenaline and the caffeine. They are already firing and discharging and so on. They have oscillations even in the control at 5 hertz. And when you gave them isopaternal, then they get all crazy. Now, we know that the Purkinjes can fire faster. They can generate the calcium oscillations with greater frequency, and they can generate greater DADs. But there's another factor that should be considered when you are looking at the firing of these events from the Purkinje versus the muscle. Both types of cells are capable of generating DADs, but the question is, which one will fire first? Well, from cellular basis, we can say, well, the Purkinjes are likely to fire first, at least from the experiments in vitro. But the cells are in vivo, and they're surrounded by an environment. The environment of the Purkinje is a linear bundle where the electrical load is relatively small. Sink versus the source relationship is pretty well balanced. Whereas in the muscle, the sink to source relationship is highly unbalanced in favor of the sink. So if the cell wants to fire a DAD, it has to recruit many, many neighbors in order to be able to fire. Whereas the Purkinje doesn't have to do that. A Purkinje cell will be easily firing without any interference with their neighbors. So we have more than one factor. We have the calcium release, we have the DADs, we have the greater calcium overload, the greater number of events, the greater frequency of events, the DADs, and the sink-to-source relationship in vivo, which make the Purkinje much more likely to fire. Yes?
Did you notice any phase 4 diastolic depolarization in the single cells in the Purkinje?
Yes, we do have phase 4, and we try to differentiate between phase 4 and DADs. In this case, for example, in the absence of high-frequency stimulation, we didn't get phase 4. Remember that phase 4 usually occurs when the cell is quiescent or when the frequency of stimulation is low.
So under normal conditions, if you don't pace, do they fire on their own?
They can fire on their own, yes, definitely. They have all the characteristics of Purkinje cells.
And the second question I had is, some people have observed a transitional cell between the myocytes and the PFs. So I was wondering if you noticed any of those kinds of cells, either when you were looking at your tissue sections where you have such beautiful, identification of the Purkinje network, or while you were isolating the cells, was there a different kind of cell?
We haven't focused on that. I did not be able to tell about that. So, DADs are there. Now, the test of fire comes in the heart. What's happening? Where is the arrhythmia generated? It's So what we did is, the first experiment that we did was to anesthetize a mouse and put give it caffeine and epinephrine, induce the bidirectional tachycardia, but introduce a needle attached to a syringe that was filled with Lugol solution. Lugol has been shown to kill Purkinje fibers. Purkinje fibers have a great amount of glycogen inside, and you can kill the Purkinje fibers without significantly damaging the ventricular wall. So we introduced the Lugol solution into the right ventricle during the bidirectional tachycardia, and we were able to convert the bidirectional tachycardia into a monomorphic tachycardia. So the alternation disappeared, and that was published in the paper by Marina Cerone. But the testifier was during the polymorphic tachycardia, and this is a polymorphic tachycardia. Notice the time, the scale. I don't know exactly. These are very rapid tachycardias in the mouse. What we did is open the right ventricle and look for the first event, the first pixel that fired at a given beat during the tachycardia. So here you have beat 1, 2, 3, 4, 5, etc. And here each color indicates the location of the first event during that beat. And if you look at each one of these two windows, each beat corresponds to the presence of a Purkinje bubble. And this one, for example, beat 1 is very clearly associated with this Purkinje in this picture. The pictures are better than shown here, but indeed, we were able to show that each one of these beats corresponds to the location of one. Now, what happens when the arrhythmia deteriorates into ventricular fibrillation? Well, the arrhythmias usually start as focal. So you have bidirectional tachycardia, which converts into a more polymorphic arrhythmia by focal events. This is one focal discharge, another focal discharge, another focal discharge. We're looking at here at the transition. A focal discharge gives rise to a wave that encounters refractory tissue here and begins to rotate. And this gives rise to a sustained reentrant arrhythmia, which is ventricular tachycardia. So what changes, the change is from a focal tachycardia into a reentrant tachycardia, which deteriorates into ventricular fibrillation. And so, let me conclude by saying that ventricular tachycardia and heart failure initiated by a focal mechanism that appears to be due to trigger activity, primarily from delayed after depolarizations whose origin is unknown. CPVT is a rare inherited arrhythmogenic disease characterized by adrenergically mediated ventricular tachycardias, leading to syncope and or sudden cardiac death in the absence of structural heart disease. Arrhythmias in the mutant mouse and by inference in the CPVT patient are likely the result of enhanced calcium release through affected channels. The mutation predisposes both Purkinje cells and ventricular myocytes to DADs and induce trigger activity. And then we can say very convincingly that bidirectional ventricular tachycardia results from the interaction between two highly localized ectopic foci, one in the right bundle branch or a branch of the right bundle branch, and the other in the left bundle branch that mutually synchronize to discharge in an alternating fashion. The conversion of bidirectional ventricular tachycardia to polymorphic and ventricular fibrillation is a result of a transition from focal to sustained reentrant activity, and this is mediated by a wave break, by unidirectional block and reentry. I do feel that this data can be useful not only for understanding the arrhythmias in the CPVT patient, but also may provide insight into patients with heart failure. We do not know what the Purkinje fibers are doing in heart failure. Nobody has ever looked at them. And therefore, I put forward the hypothesis that in heart failure, arrhythmias, due to focal activation, may be initiated in the ventricular cells. So let me finish by just acknowledging some of the people that worked. First of all, Sylvia was a great collaborator. She gave us the mouse, and she sent me a postdoctoral fellow. So what else can you ask for? And this was Marina Cerrone, who now not only was my postdoctoral fellow, but married my partner, so Mario Delmar. So now Marina and Mario are happily married. And then you have Sammy, who actually is our mouse optical mapping expert. Elena Tolkachova, who did some of the single cell recordings in Purkinje cells. This was on the direction of Justus and Umanuel. Sergey is our optical mapping guru. Karen Bikstrom is our molecular biologist guru. Omer Berenfeld is our mathematical modeling guru. Sandy Patrick is yet another guru, really from India. This is Todd Heron, who did all the calcium imaging collaboration with Michelle. Michelle is a postdoc in the laboratory, and she collaborates with both Todd and my group. And of course, we are supported by the NIH. So I want to thank you for your attention. I'd be happy to answer questions.
So I'll start. So you have to reach, so this is a stochastic process. The release of consequence from the random receptor is a stochastic process. It really is a probabilistic process. They have to get all of those random receptors get to a situation that they leak at the same time and so on. And that by itself requires some synchrony of stochastic processes. Then you have 1 year and 1 year. And you're telling me they're synchronized, and this fires once, and this one knows how to wait, and then each fire once, and then each fire once. That's tremendous. How does it happen?
Well, I've asked myself that question. I don't know how it happens, really. But as you know me for what, 40
Long
years?
time we wouldn't get into
We wouldn't get into that.
You know, it's up to the gurus.
When I was a young man, a few years ago, and I was a postdoctoral fellow in the laboratory of Gordon Lowe, I began studying coupled oscillators. And I began studying coupled oscillators because I was interested in parasystole. As you know, parasystole is an arrhythmia where there's an ectopic focus in the ventricles that fires independent of the ventricular discharge, and is protected somehow by the surrounding tissues. And when the ventricles are not refractory, there will be a premature discharge, and traditionally has been associated with variable coupling intervals between the normal rhythm and the ectopic discharge. But at that time, Gordon and I demonstrated that Purkinje cells can be entrained by subthreshold depolarizations in their surroundings in such a way that if a Purkinje fiber has pacemaker activity, and a subthreshold stimulus occurs early in the cycle during diastole, it will delay the next discharge. And if it occurs later in the cycle, it will advance the next discharge. And you can build a phase response curve in which there will be an initial phase of delay followed by a phase of acceleration. And using that phase response curve, we provided a mathematical model that described very nicely the patterns of excitation that could occur in a patient with parasystole, even those with variable intervals. And we demonstrated that depended on the basic relationship between the normal heart rhythm and the ectopic rhythm. Then I went on to use the same idea to study interactions between pacemakers and the sinus node. And using a very old preparation called the sucrose gap, I introduced a slice of the sinus node. I just made slice with a Gillette blade and caught the sinus, and the sinus node bundle was beating. And I placed it into this 3-compartment tissue bag that was divided into 3 compartments by 2 rubber membranes that had holes in the middle. So the bundle was threaded through these bundle, and then I superfused the central compartment with sucrose solution in the absence of any ions, and that insulated the 2 outside pacemakers. And lo and behold, when isolated, each one was beating at its own rate, and they were not synchronized. But then I put a salt bridge between the 2 compartments, jumping the sucrose gap, and lo and behold, they started synchronizing. And then I did experiments showing that indeed the 2 pacemakers could synchronize each other, could mutually entrain through their basic relationships in such a way that if the faster discharge occurs early in the cycle of the slower discharge occurs early in the cycle of the faster one, it would delay the next discharge, and vice versa. So the synchronization of the pacemaker is an entrainment, a mutual entrainment. It's a democratic process where the slower and the faster ones will entrain. Let me.
That I understand. And I know those studies, but I maybe misunderstood my point.
No.
The point is the concept, not the
Right.
entrainment of the pacemaker once there is membrane excitation. The calcium cycling is stochastic. The
Right.
release from the renders is stochastic.
Right.
And it has no long distance communication.
Absolutely.
So, the
But let me
generation
finish.
of this thing, it depends on
But it's
the-
averaging across a lot of cells that are in the Purkinje, and then it'll be relatively uniform.
Yoram, let me finish. So you accept the first premise, right? The premise of mutual entrainment. Now, just remember that both types of cells are receiving stimulation from their surroundings, okay? So each one is triggering a discharge. And it can happen under certain conditions that one discharge can trigger the discharge of the other one. And that can lead to the synchronous, not because they're very close or very far away, but because of the discharge, the relationship between the latency of the DAD and the previous discharge. That latency in itself and the amplitude of the DAD is the one that are going to decide whether this guy is going to trigger the activity of this one and vice versa. Now, I have no way of proving that right now, but I think it's a feasible hypothesis.
So is that your idea in the end, that this one fires,
Yes.
and that's what drives everything, and its DAD then is what triggers the next
That is
one?
DAD,
to fire?
the DAD of the other one.
Yeah, the DAD from this one triggers the beat of this one.
Exactly.
And then the
But they-
DAD from this one triggers that.
Triggers the beat.
So does this make, so in other words, this is going back and forth across the disc bundle?
I think it's going back and forth across the ventricles.
But if the mechanism of the generation of the DAD is spontaneous release of calcium through the random receptor.
So then does that make a prediction about what's happening in
But-
the
but
atrium?
it's
that you can
necessary-
measure?
I'm going back to the source of the DAD generation, which is the Ryandian receptor leak.
What is necessary is that there is triggered activity, that DAD leads to triggered activity. If there's no trigger activity, there's not going to be synchronization.
But if there is no synchronous generation of the DAD through synchronization of these stochastic release processes for the Ryandian receptor, then you don't get.
But they're not
DAD.
totally stochastic, because
No,
they're triggered by
right
the
now they're not
action sensor of the-
stochastic because they're being triggered. You are now under conditions.
We'll talk later.
You are now under conditions in which DADs are going to form, regardless of anything
So they
else.
form anywhere.
But DADs are not
They-
totally
they can
random.
form anywhere.
They
But
only
when you have
expressed
beats.
in those locations.
They express primarily in those locations because those are the ones that have
Because of the
more
entrainment.
calcium overload.
That's right.
Yes.
But the calcium overload is everywhere in all Purkinje cells.
You know Purkinje cell,
And
but
they
that-
all generate spontaneous releases.
Right, but what I'm trying to say is that perhaps it's not just one Purkinje cell, it's a group of Purkinje cells that fire together.
DADs are synchronized by the action
Right.
tentacles. If you measure in Purkinje fiber,
Yes.
you get
Uh-huh.
synchronized DADs.
So I wanted to actually follow to what Colin said. So actually it might be alternative explanation. So it could be a junctional tachycardia. What we found evidence already in 2 species, in humans and in rabbit. as you go from AV node into the his bundle, there are two parallel compartments. And they do, it's known already for probably 40 years that there is longitudinal dissociation.
Right.
So essentially you may have, in a situation like this mutation, a junctional tachycardia, which just exits to the left, to the right, and then entrainment through his
Yes,
bundle.
there are several hypotheses. I didn't go through all of the hypotheses. The first hypothesis is not a junctional tachycardia, but it has to be below the junction. And that was put forth originally by Doug Zaps, actually, said that there could be a unidirectional block alternating with these mutations. And that could happen.
No.
Or, yeah. A unidirectional block, junctional tachycardia or his bundle tachycardia with unidirectional block alternating. Otherwise, you will not have it. You will need a unidirectional block.
So your presentation, essentially is sort of Art Winfrey-like in the sense of the properties of excitable media, in my mind, and it leaves out any electromechanical or mechanoelectric stuff. And well, the reason I ask is because of your point, which is how does the thing over here know what's going on over there? Well, if there's mechanoelectrical, electromechanical feedback, the depolarization here causes mechanical activation of the tissue over there, which could set up all sorts of stresses and strains and depolarize not just the membrane, but it could fibroblasts, if you like, fibroblasts to be electrically active.
Or you just got some
See if that gets, see if Levy 7 gets through it.
I think the mechanical event is feasible. That's an alternative hypothesis that I think is could be tested. But the fibroblasts, how would they mechanically become?
Well, the fibroblasts, according to Cole and others, fibroblasts are electrically active. And if you stretch them, they depolarize.
Well,
I'm just mentioning what I read, it's not what I do. But it's intriguing that there are strain gauges.
Right.
There are strain gauges all over the heart, and you know, as an invasive cardiologist, we know there are strain gauges because if you touch the endocardium with a catheter, you immediately depolarize it just by touching it.
I've stretched Purkinje fibers to their limit. Okay, and they like it much more than ventricular myocytes. Ventricular myocytes don't like to be stretched. Purkinje fibers don't mind being
They don't mind.
stretched. They don't mind being stretched. They don't have the deep tubulars, and the membrane seems to be more flexible. I can assure you that Purkinje, stretching a Purkinje fiber to its limit is not a thing. So possibly it may happen, but we need to test that hypothesis. The alternating junctional tachycardia is a possibility that we don't know. But these animals do not present bundle branch block. They don't have any reason to have bundle branch block.
I will write to you a detailed explanation. It's a little bit different.
Junctional tachycardia, if it is a junctional tachycardia, it has to block on one side or the other.
No.
No?
So we find evidence in humans, and there is also evidence in rabbits. There are two compartments. They express different connections and probably different ion channels. And in some conditions, they don't talk to each other at all. So you may have essentially excitation within his bundle, which excites on the left side or on the right side under some conditions. But they can still talk.
Under what conditions?
Well, that's exactly what you have to determine, not me. But the simple question I have, did you try to put Charlie Birul's catheter in those mice during tachycardia? So and look at his bundle electrogram.
Well, we looked at his bundle firing during the action. We have a map of his bundle firing. We don't need the Charlie Birul.
Can you see retrograde atrial activity during that bidirectional tachycardia?
No, I haven't. It's possible, but I haven't looked at it.
Which would give you an idea of what's going back up through the bundle.
Through the junction.
Yeah.
Heavy junction.
Yeah, transitional cells.
Yeah. So what are the conditions in which you get that?
I can give a talk for 1 hour.
Anyway,
Other questions? No other questions? Comments? Wishes?
Yes, yes, yes. Go ahead.
How different is the calcitonin to ryanodine receptor interaction in the Purkinje cell?
We don't know.
and the
Actually,
myocyte?
Purkinje seemed to be, Penny Boyden published a paper a couple of years ago in which she studied the calcium release mechanism in canine Purkinje cells. And canine Purkinje cells have the RYR2, they have the RYR3, and the IP3 receptor, which makes things very different from, since Purkinje cells do not have the T tubules, they have to utilize a different mechanism from the myocytes. And she demonstrated that the release of calcium, the sparks in the Purkinjes, occur primarily in the center of the cell from the RYR2, not near the T tubules. Okay, so we don't know in the mouse what's happening yet. We have done just whole cell calcium imaging, but we haven't really looked at the sparks carefully or any of that. And we don't know if the mouse Purkinje has IP3, active IP3 that is doing something important for the release. We don't know if the RYR3 is important.
Calcitonin can play some role in determining the refractory state
Of the calcium,
the
sure, absolutely.
ryanodine receptor.
I don't know. I don't know what's happening in the Purkinjes, but that's a good question. And there's a calsequestrin knockout mouse that has a very similar phenotype with bidirectional tachycardia, so it could be studied. That question, but as of now, no. Any other questions?
Thank you very much again.